morning with breaking news. We have news that 4 dangerous mutations have escaped from prison. One of the mutations has changed a C in the DNA coding strand to a G. We are going to go over to Karen for an explanation of how this will affect our translated amino acid sequence. Hello, and thank you, Caitlin. We have identified the mutation who has done this as the Missense mutation. We know this because the change in bases has caused a change in the amino acid. Here we see that the C in AAC was changed to a G. This in turn changed the amino acid from asparaginine to lysine. We are unfortunately a bit worried about this as it could potentially alter protein function or even just make it non-functional. We also know that we are now dealing with a transversion as the base was changed from a pyrimidine to a purine. Unfortunately, while Karen was on air, we received news that another mutation has attacked. It looks as though this mutation has changed the TGC in the DNA coding strand to TGA. This will also affect the translated mRNA, changing it to UGA. This means that the amino acid sequence will be changed from cysteine to a stop codon. We are now also dealing with a nonsense mutation. We know this due to the fact that the amino acid cysteine was changed to a stop codon. It is also clear that this mutation is a transversion, as the base was changed from a pyrimidine to a purine. Now we're going to take it over to Bruce from the Genetics Police Department, who has just announced that he has made the arrest of a 3rd mutation.
Thank you, Caitlin. As we are monitoring the DNA coding strand for any new developments, we noticed that the C in CCC was changed to a G, meaning that we now had CCG. We are lucky to have caught this mutation when we did, as he is a difficult one to find due to the fact he is a silent mutation.
So you're saying that he's difficult to find because his changed codon still codes for the same amino acid in our translated amino acid sequence?
Yes, exactly, Caitlin.
See that the CCC that was here has now been changed to CCG, both of which code for proline.
We are dealing with a transversion here as a pyrimidine was changed to a purine.
The Genetics Police Department has just let us know that they have identified the 4th and final mutation as read-through. They have discovered that the read-through mutation plans to change the TGA in the DNA coding sequence to TGG. They were able to identify this mutation as read-through due to the fact that the amino acid in the translated amino acid sequence would change from a stop codon to tryptophan. As you can see here, the A is replaced with a G in both the DNA sequence and the translated mRNA, which causes the stop codon to change to tryptophan. This mutation would result in a longer protein. We can see that this mutation would be a transition as a purine, the A, would be changed to another purine, to G. The 4th and final mutation has been caught. We can now relax as the repair mechanism teams move in to repair these mutations. Thanks for watching, folks, and we'll see you next time on Genetics Daily.